So, remember, we've been going through this series on research validity, which is a study has research validity when you get to the end of it and you make accurate conclusions based on your research about what's going on, the relationships between your variables. And there was 4 pieces to that. There was statistical validity, which was minimizing your type 1 and 2 errors. Internal validity, which was reducing sources of confounding. There's construct validity for causal conclusions, which was operationally defining your variables in ways that are valid and reliable. That's measuring and manipulating things. And we got the final one tonight. That is external validity. It's when your study findings generalize to other samples, settings, procedures, variations in how you manipulate or measure things, and times. So we call that external validity. And that's basically when your findings are replicable across variations and procedure and whatnot. So let's dive in. It's not too bad of a topic. So again, external validity is when your findings generalize to other sample settings, procedures, manipulations, measures, times, etc. And how well do your study findings actually apply to the real world? That's the question being answered by external validity. And so if they if your findings are real, so they are a truth you've discovered about human nature, they should generalize to the population beyond just the sample in your study, right? They should also generalize to other places, other situations, so variations in methods, conditions, variations in how you define the IV, and outcomes, that is variations in the DV. So that's setting conditions and outcomes. And finally, your findings should generalize, if they're truth, across times. So should be more than just the people at one time. It should generalize to the past and the future. So replication is how you know if your study has external validity. So you're not really going to know based on your dissertation whether you've captured one of the laws of human behavior. It's just sort of other people following up. But one way to get a dissertation done fast is by doing a replication of someone's prior work and just sort of doing a variation on it, which we'll talk about on the last slide. So external validity with regard to participants is when your findings should generalize to a population beyond just your study sample. So believe it or not, we're not really interested in whether there's a relationship between the variables and just the people in your study per se, right? So we want to know, not really, I mean, it's great and all that there's a relationship in your sample, but we're trying to identify things that are going on in the larger population of humans, right? So if your study results only apply to the sample, you obviously did not discover some sort of deep truth about people in general, so why should anyone care? So your sample selection is the key to your ability to generalize your findings to a larger population, and we just talked about this earlier in the lectures. So if only college students are used in a study, you really think you're going to be able to generalize your findings to other non-college students? I mean, maybe. Some findings will, some won't, right? So there's cohort differences, for example. Like college students tend to be younger, so using college students for research may not generalize to a larger population, but some things are, big enough effects that they will, or they're common to all people. What if you have mostly women or mostly men in your sample? Can you really generalize your findings to both sexes? Again, it depends. Some things will generalize, some won't, but it does reduce your external validity for participants. So how do we know whether our findings will generalize to other participants? Well, we replicate, or other people replicate, right? So we repeat the study with different types of participants, and that's really the only way you know. So one way to build in external validity for participants is doing what we call sampling for heterogeneity. So you improve the likely external validity of your results by getting a diverse sample in the first place. So you build in sampling variability. So ensuring that the sample is representative of that larger population makes it more likely, right? Remember representativeness, that you're going to be able to generalize to the larger population. So sampling to ensure that both men and women are adequately represented in your study is a great way to be able to increase the likelihood you're going to be able to generalize your results to both men and women. So, a lot of drugs, for example, they only test them on adults. So for teenagers, they don't know the effects. So they off-label, put people on drugs that haven't been tested on kids. And so that's actually fairly common. but it is a problem. You don't actually know what's going to happen. You don't know if the results for adults generalize to children. So sampling for heterogeneity may improve your external validity, but it may decrease your statistical validity, because when you got a more diverse group, you've got just sort of more noise, more between group differences in people, and so it makes it harder to reject null hypotheses, for example. You get more noise. So another thing that can happen is when you have a diverse sample is the subgroups may differ on some effect or an IV effect, and those they end up canceling each other out. So overall, you say there's no effect when in reality there's subgroup effects. So for example, men might have some positive correlation for two variables, and women might have a negative correlation. When you add them together, you end up with no relationship whatsoever. So you need to, where you suspect that, you need to make sure you have enough statistical power, that is enough n sample size, in order to look at the subgroups separately. So non-random sampling does not necessarily mean you don't have external validity. So remember, that was one of the methods we talked about in our earlier lecture, where you don't do things randomly. And just because it's not a random sample doesn't necessarily mean your results won't apply to other participants. So introductory psychology students, they do have the same eyes, memory, and so forth as the general population. However, studies on eyes and memory and whatnot might generalize, but they might not be representative of the general population when it comes to other things like IQ and attitudes or social issues, etc. So it depends on the topic sometimes whether your results will generalize. So it depends on the topic. So volunteers are a problem we all face because you can't force people to be in your study. So the fact that we only use volunteers for research can be a problem with your external validity. Why is that? Well, people who volunteer tend to be nicer, tend to be more in need of social approval, etc. They also may have particular motives. The opposite could be true. So again, bring up who fills out the customer cards at restaurants. Is that a sample? Can you generalize that to all customers? They're generally people who are really mad or really happy, right? It's not a true sample of the whole range of things, just to the endpoints. So when it comes to demographic groups, your findings should generalize across different demographic groups unless only one demographic group is of interest, right? So be careful not to define your demographic groups too broadly or too narrowly. I see this all the time. For example, people categorize folks as Asian, and Asian is a really broad category that ignores lots of cultural differences between people. So another one I see all the time is they just say basically anyone under 18 is a child, and that's just kind of goofy. There's huge differences in development between a 10-year-old and an 18-year-old, right? And they just ignores that by throwing everyone in a group, and I think it's kind of ridiculous that they do that sometimes. So differences in cultural beliefs may limit your ability to generalize to other groups. So for example, research on Asian women may not generalize to African American women. We actually have to adjust different ethnic groups differently in our hospital surveys because some groups tend to be harsher, some groups tend to be nicer, and so we actually have adjustments for that we do because it's a known sort of cultural beliefs differ. So again, replication of findings among different groups is your key to establishing that you have external validity of your findings or results across participants. So cross-cultural studies are particularly powerful ways of identifying actual laws of human behavior when they can show an effect is consistent across different cultures. Like, for example, symmetry as a sign of beauty is something that is seen cross-culturally. It's almost universal as a sign of beauty. So it really makes you think, maybe this is one of those true sorts of laws of human behavior. Symmetry somehow is viewed biologically as an attractive trait in a partner. So when it comes to using other species, so that can obviously limit your external validity if you're doing research on rats or whatever, but not always, right? So Pavlov's work with conditioning, it worked on dogs. That's where he found it. That's the bell and the salivation stuff. But it also works in people. So not always, but there's definitely some issues to consider there for using other species. So there's also external validity for settings or conditions or outcomes. And basically, your findings should generalize to other places, situations, variations in methods, variations in how you manipulate the IV, and variations in how you measure the DV. So this one's actually particularly important. And this is where you can get a dissertation, by the way, pretty easy. So findings that have high external validity can be replicated across variations in places. So for example, and this particularly useful, like if you're doing effectiveness analysis of programs, right? Program evaluation. So do laws that are shown to be effective in California work when they're used in different states? Well, if the law is truly an effective law, you would think it would. How about situations? That is sort of variations in methodology. So do altruistic behaviors work the same in group versus individual settings? Nope. But that led to some additional information about diffusion of responsibility and those sorts of things, how people are altruistic individually versus in groups. And how about conditions? So you slight variations in the DV. So if I vary my operational definition of my IV, so I'll use pictures of the crime scene versus videos of the crime scene. That's variation. Do I see differences in the outcome? I don't know, but to the extent that I see consistent findings across variations, it suggests my results are more externally valid, right? And the same thing for the DV. So, do I get the same results when I use the Wechsler intelligence scale versus Stanford-Binet? Do I get wildly different ones? Why would that be the case? So these are all like variations in, place, the methods, how you define the IV, and how you measure the DV. And things that are true laws of human behavior should work across variations in these things. So volunteer organizations are a problem just like volunteers overall. So organizations that volunteer to be in your study may differ from others, right? Which causes problems with external validity. And this actually happens a lot. Like when an organization volunteers, they're motivated. So let's say you got a school that volunteers to test some new reading program. I mean, you might get fantastic results that you can't replicate with other schools because those other schools are less motivated. And so that's the problem with volunteer organizations. It's basically the same as the problem with volunteers, period, right? Less motives. So interesting, and something folks confuse all the time is the difference between efficacy and effectiveness. So results found under ideal experimental conditions don't always translate to real world conditions. And that's the difference between efficacy and effectiveness. So what are they? So efficacy is results that you get under ideal conditions. So like scientifically controlled conditions. If you find some drug works or whatever, that's your efficacy. Effectiveness is the results you get under real world conditions. So less controlled conditions in the real world, do you get the same results? And some things are very efficacious but not effective. So for example, methadone treatment was found to be very efficacious for heroin replacement therapy when it was used in randomized controlled trials. When there was tight monitoring, they made sure everyone took their drugs, etc. When they went to neighborhood clinics and tried using methadone treatment, it was much less effective, and they actually had to put additional controls on there because it turned out it had a street value, etc. People were ODing, all sorts of crazy stuff. So it was it had high efficacy and less effectiveness, and they actually pulled back and like they make people come in and take their pills and things like that to try to get closer to those experimental or efficacy results. Anyway, that's the difference between efficacy and effectiveness. So laboratory experiments are frequently criticized because they're kind of artificial, right? Like, they're all controlled and they're artificial, and they might therefore lack what we call ecological validity. So ecological validity is when your findings generalize to the real world. So going from the lab to the real world doesn't always work. It's the same problem with efficaciousness versus effectiveness. So phenomena that are studied in the laboratory should work outside of it, right? You'd think. So antidepressants that you develop in a lab or political ads, they should work outside the lab as well, or what use are they? So the way you figure this out is you do field research, like they did with methadone, for example, to determine the external validity, whether you've got ecological validity of your results. And there is sort of this balance between internal validity and external validity because you get more control in a lab, but you lack your ability to generalize your results outside of your little controlled experimental lab. So for example, labs provide more control and hence you end up with better internal validity for your results. If you get to the end, if you see differences, it's due to the drug or whatever it was you were manipulating. But they're artificial, which can limit your external validity. There's also pretest sensitization, which we've talked about before in the internal validity chapter. So repeatedly measuring the DV not only affects your internal validity, but it can also negatively impact your external validity. Again, being asked to keep a diary of your angry thoughts, and that's the DV for a study, it can have more impact on your subsequent angry thoughts than the anger management, right? Treatment. So, your results can be influenced in ways you don't expect from sensitizing folks to the dependent measure. So finally, we've got external validity for time. So findings should generalize to other times. High external validity results generalize to both the past and the future. So history confounds, again, this is a repeat from the internal validity lecture, is when study findings are specific to a specific period of time because of events surrounding that time. And it's when the IV is confounded with other events that occur in time. So work programs, for example, are probably particularly effective during bad economic times, but less effective during good economic times, right? Because folks don't need them as much. There's also generational confounds, and this is when you get results that are specific to a particular generation because of idiosyncrasies about that generation. So like some of my favorite examples, those scary driver's ed videos that they used to show in the 60s, they probably make kids laugh today, right? They're corny. And other interventions, for example, Reefer Madness is actually a musical now, so my guess is it's not going to be particularly effective with the current generation for reducing marijuana use, right? So replication, finally, is our key to knowing whether your study has external validity. That's knowing whether your results stand across variations in participants, settings, times. You got to replicate it. So no single study proves anything, just to repeat from very early in the course. Only results that are repeated across participants, variations, settings, and times are considered truth. That's how we know what's real in terms of effects. And they establish when a relationship exists and also, variations when it doesn't exist, when things don't work. So does it work in cities but not the country? Does some intervention work at nights but not during the day? Does it work for young children but not adults? These are all established by doing follow-up studies and replicating things with variations. So you can do, and this is where I promised there'd be some advice for doing your dissertation. You can do exact replication, and exact replication is when you repeat the exact methods of some other study, and they usually won't let you do that for a dissertation because you're just repeating what they did. Plus, it doesn't really add to the literature except very much, right? So what they really want you to do is what's called a conceptual replication. This is where you kind of repeat what someone did in a prior study, but you vary something. So you actually test the external validity of the findings. So you'll use slightly different procedures or different methods for the independent or dependent variable, different sample, etc. So use study stories instead of videos to study propaganda, for example. That would be, you're still replicating the idea, but you're changing things a little. That is an easy dissertation. And then finally, just to bring it back up again, literature reviews, but particularly meta-analyses, are excellent summaries of sort of the state of the knowledge for particular topics. So whether there's research validity of a particular area of study, look to your lit reviews, and in particular meta-analyses, which are those mathematical lit reviews. So that's kind of all I got on research validity, and overall, hopefully this was useful for when you're setting up your dissertation. Post any questions to questions, and otherwise, that's it.